So tonight we're going to be talking about concepts of what disease actually is, and in contrast, what health actually is. We talked about humoral theory, numeral theory of disease, but we're going to go into a little more detail on that. Just to remind us where we're coming from, at the time we got to the Renaissance, most people agreed that the humors and their imbalances were what the cause of disease were. But people had noticed that disease doesn't just come out of nowhere. It tends to spread from person to person, tends to happen at certain times of the year, and there was really no definite way that they were able to model what was causing that. So people made progress, especially in the Islamic empires, in categorizing and distinguishing one disease from another, but the actual cause of disease was still very much up in the air. Miasma, or bad air, was a very common and popular idea that bad air caused you to get sick. Contagion, which is just basically the observation that disease can spread from one person to another, but through means that no one really understood. Spiritual judgment, the idea that you were being punished, or you were being punished for the acts of someone in your past, a family member, still held on as a cause for disease. Humoral imbalance, very popular idea. You have too much blood. What's the solution? We take it out. You have too much black bile. We probably take out blood for that too. That was the real popular method. And then diet, something that actually holds up, that your diet can actually be the cause of certain disease states. So we finished last time talking about how anatomy became an actual science because of Andreas Vesalius, basically used experiment and recording his findings to document how things actually were in the human body, and from there, the idea that you could document the changes that occurred in disease took hold. Now, one thing I didn't drive home about Vesalius is that unlike Galen, he wrote that if you didn't believe what he had to say, you could just do it yourself, and he gave instructions for that. So what happened in the 1500s, and hopefully is continuing up to this day, is the idea that it's not good enough to rely on authority. You should be able to go out and prove these things for yourself by repeated experimentation or repeated observation. So he worked on modern anatomy and then retired to be the court physician for the Holy Roman Emperor before dying in Greece. The next person who really brought up the idea of the causes of disease was another Italian, Girolamo Fracastoro, and he had a really modern idea of what disease was. He thought there were little seminella, or seeds of disease, seeds of disease that could spread from person to person, but no one really took this seriously, no one investigated it. So we had an opportunity for a more modern idea about how disease spreads happen to take root, but it didn't. He actually became more famous for naming the disease syphilis. And for you, those of you out there who want to make a name for yourselves, try to take this one. Don't just write up your findings in a medical journal, write it in an epic poem, because that's where syphilis comes from. He wrote a poem describing the progression of the disease syphilis, and that it was caused by a curse from Apollo onto a shepherd named Syphilis, and that's where it came from. But in the process of writing this poem, he laid down a very accurate description of the disease and its progression and its effect on the body. Another early pathologist was named Jean Fresnel, and we're shifting now towards France, and that's going to be a theme in pathology, is that we have different centers of investigation taking precedence from one another. So he did another reorganization of disease and how it works, and he looked into how the physiology, the pathology, and therapeutics could work as individual topics, and he come up with a new classification of disease. Now, his classification was not something that we today would look at as being all that worthwhile. He divided diseases into ones above the head, below the head, above the abdomen, below the abdomen, ones that caused dryness, ones that caused wetness, and looked at them as either generalized or specialized. But the process of reorganizing these diseases is where a lot of progress was made. Sometimes it's not about getting the answer right, but starting the process of questioning. So just a quick jump forward, if you can think about Sigmund Freud, most of his theories have not held up over time, but by getting the ball rolling in terms of looking at psychology and psychotherapy, some good things came as a result. Not all good, but it's sometimes a matter of the rolling of the process leads to the big breakthroughs. Another breakthrough was actually looking at the human body in the manner that Vesalius did, but looking not for healthy, normal tissues, but looking for things that had been damaged. And the big name for that was another Italian. This is Marco Aurelio Severino, And he was looking at the diseases and afflictions in the people that would come into his practice. Now, the interesting thing here is that he had the exact opposite issue that we have in contemporary medical schools. We get to see people who have passed away and donated their bodies after a long lifetime and have probably got different diseases at different stages of progression, and we get to learn from that pathology. And in the lab, I'm sure everyone by now has seen more than a few things that are not strictly from the atlas, but are actually examples of different afflictions this person had. Back in these days, most of the people that were dissected were young, healthy men because they were all people who'd been condemned to death for different crimes. And his quote is just interesting in that it's the exact opposite issue we have now. If we had one complaint to lodge, it would be that a lot of our donors have very poor muscle tone and atrophy of the muscles because of their age. Whereas back then, they had nothing but healthy young people to dissect who had no evidence of pathology, and they had to wait to find people with pathology that they could actually investigate. Now, another big name, both in physiology and medicine, is Franciscus Sylvius. Now, he was a really staunch advocate of medical progress. He really championed William Harvey's idea that the blood circulates, which is one reason it actually got accepted. Harvey had some really amazing observations, wrote them down, but he didn't draw a great deal of attention to them. In fact, his major contribution about the blood circulation is kind of tucked away in the 10th chapter of his book. But people like Sylvius made sure that people took note and started debunking Galenic problems that had developed over time. So he worked on chemistry and how the chemistry in the body actually could be the cause of disease or health. And he thought that there were alkaline and acidic elements that were normally at balance in the body, and if they got disturbed, it would cause disease. And yeah. So basically, our body has a very rigid control over how we keep ourselves from getting too acidic or too alkaline. Our lungs and our kidneys do a great job of preventing that from happening. Have you guys talked about metabolic and respiratory alkalosis and acidosis yet? All first years, you will be banging your head against it shortly, so you're welcome. Now, we talked a bit about William Harvey, but the big problem for Harvey was that he had to theorize that there was a connection between the arteries and veins that was simply too small to see. And the first person who was really able to make that observation was called Marcello Malfigi, and he was one of the early microscopists. Now, microscopy initially was very difficult because the lenses they used didn't correct for different wavelengths of light, so it would tend to scatter according to the different wavelengths. It was called chromatic aberration, so reds, greens, blues, purples would scatter to a greater or lesser degree because they had different wavelengths. But they were able to make some decent observations of frogs' lungs, the capillaries therein, and he was able to draw and demonstrate that there were literal connections from the arteries and veins, basically backing up and authenticating William Harvey's circulation of the blood. The pathologists at the time were largely people, they weren't called pathologists, they were hospital physicians who would treat patients, see what they came in with, and then do an autopsy at the time of death. And autopsies became more standard in the 15 and 1600s, which is why pathology actually started to become an eventual science. Now, the first real compendium was called, has a great name, Sepulchridum Sive Anatomica Practica ex Cadaveribus Morbo Dentatis, or the pathology seen in the dead body. The problem with this one was it was a good compendium, but all the cases were just listed with no real organization. It was just one case, then another, and it wasn't terribly useful unless you'd gone and indexed it yourself. The first person who's kind of considered to be I want to say modern pathologist, but someone who started the actual role of modern pathology grossly was yet another Italian, Giovanni Battista Morgagni, and he published his own compendium of pathologic disease. And it was basically a lifetime's work. He worked in a hospital, and he would see the patients come in, he would track their progress, track what would happen if they died, he would then do the dissection and autopsy, and document the problems. And what he found was that these people did not die of too much black bile. They didn't die of an excess of phlegm. They died because they had tumors in their organs. They had lacerations. They had hemorrhages, and they were affecting distinct organs. And he said that the seat and cause of disease, which is the name of his book, De Sedibus et Causibus Mulborum, was basically the organs. They were the places that disease happened that killed us. And this was really the first time that humoral theory was being challenged in a way that it couldn't respond to. Now, authoritative sources hang on for a long time, and there were people promoting humoral theory up into the late 1800s. But this was the first time people were really seeing that there were definite problems with it that couldn't be ignored or explained away. Now, when you think about the French Revolution, I'm assuming most of you are thinking of, what's the big famous musical? Les Miserables, there we go. If you ever get a chance, go on YouTube and look at people complaining about it, because apparently they did everything wrong when recording the music for that, like everybody was stressed out and dehydrated when they were trying to sing. Anyhow. The main idea with the French Revolution is it completely upended French society. French society had been very hierarchical, very structured, very rigid, and very much difficult for anyone to make any sort of innovation take hold. The revolution happened, and suddenly it was flipped the opposite. Old institutions were eradicated, old distinctions were eradicated, and suddenly new, talented people could get to work. That was maybe the most beneficial thing from the French Revolution. It was very destructive, caused a lot of trouble, and like many revolutions, got out of hand and started feeding on the people who'd been a part of it just as much as the people who were perceived as the problem. And in fact, several scientists were put to death, including Antoine Lavoisier, during the Reign of Terror. But if we're going to look for a silver lining, one thing that did come about is that French universities, medicine, and academia were much more willing to investigate new ways of doing things. And that's exemplified by French pathology. Pathology and the study of disease in France really took a massive jumpstart after the revolution, because so many new ideas were being embraced and not held back by the status quo. So two guys in particular, Marie-Françoise Xavier Bichat and Jean Cruvallier, started looking at diseased organs under the microscope. So we had Morgagni saying that organs were the seat of disease, but Bichat and Cruvallier would investigate the organs and find not every tissue in every organ was diseased. Sometimes it was just one part of an organ. And on the right, we can see that we've got pericarditis, we've got the pericardium and visceral pericardium of the heart inflamed and irritated, but they would find that the muscle of the heart, not so much. It was doing okay. So they were getting the idea that there were actual tissue changes at the root of pathology, not necessarily entire organs. Further and further away from that humoral theory. Now, we're going to pick up the idea of contagion now, and miasmas and bad air, and talk about the Broad Street pump and the cholera epidemic in London at the time. So a guy who we'll talk more about as a surgical pioneer, Dr. John Snow, this guy. That John Snow knows nothing. And he knows some good stuff. So, John Snow was a physician, and he did what was considered to be the first modern investigation of how disease is spreading, and actually looking at how it would move from place to place. Now, at the time, the area around London that was having the major cholera outbreak was close to the Thames. The Thames was incredibly polluted, and it was just presumed that miasmas and bad air coming off the water were causing the disease, because people further up on the hill with more fresh air tended not to have the disease break out where they were at. Snow went from house to house and traced the kind of contagion of people in one area. And he really came down with the idea that not only was it localized in an area, it was localized around a single water pump. And after investigating it, he found that the cesspool for a house next to the water pump, where people had died of cholera, was leaking into the drinking water of the pump. So these people were basically drinking the fecal matter, infected fecal matter of the people who'd been in that house. So he interceded, stopped the outbreak from going further, but it's actually thought that he stopped a worse outbreak from happening because he stopped that well from being used when some other people in the nearby area were having another cholera outbreak. And in terms of just pure disgusting visualization, they said that the bricks in this well were just basically oozing gunk through the seams, and you could kind of just pull the bricks out and in because of all the liquid that was moving across. Ew. So essentially, he showed that these outbreaks were not due to miasmas, they were due to bad water. And what was really interesting is there was a clergyman who had the complete opposite idea and was debating with him and came around to fully embrace Snow's ideas because he was brought around by the weight of the evidence. In fact, Snow was able to show that an outbreak in one of the higher status areas of London, far away, was caused by people getting water from that same infected pump because they typically thought it tasted better, and they actually brought it into a different area, and that caused another localized cholera outbreak, which would have been completely inexplicable by the other theories of disease. So we'll come back to John Snow. He did a few good things, both in surgery and in epidemiology. Now, after France, Vienna became the real prime location for medical innovation, microscopy, histology, pathology, and so forth. Vienna had a massive hospital open up called the Allgemeines Krankenhaus, or kind of general hospital for Vienna. And we're talking about 14,000 patients annually. So we'd have a massive number of patients coming in, all of them having their disease checked, tracked, tabulated, autopsies done on everyone who passes away to see what happens with the disease. So we've got, not dismissively putting it this way, but it really is almost an assembly line of medical investigation to figure out how disease spreads. Sounds like a great thing. Problem is, there's a problem with that approach that became more evident as time went by. Now, the person who put this all together and one of the early pioneers of Austrian pathology is called Karl Rokitansky. And my absolute favorite bit of medical trivia is Karl Rokitansky is the inspiration for Mad Max Rokitansky. So if you've seen any of the Mad Max movies, either Mel Gibson or Tom Hardy, his name's Rokitansky. The guy who directed and wrote all the Mad Max movies, George Miller, is actually a physician, and he bankrolled the first Mad Max movie by pulling extra shifts in the ER to pay for the movie as it went. And there's little anatomical, surgical details scattered throughout Mad Max, like pneumothoraxes, blown pupils, and stuff like that. So if you're looking for an action movie break, but want to convince yourself you're still studying, you can pull it off with a Mad Max binge. So we've got this great machine for investigating pathology. The problem is there was no understanding of how disease spreads and actual germ theory. It was not a thing at this time. We'd had the idea that there were seeds of disease, but it was dismissed. So as this teaching hospital proceeded through its work, it was noted that the obstetrics ward had a roughly 14% fatality rate when it was staffed by physicians. But the same process was taking place in another ward by midwives and had roughly a 3% mortality. And the mortality was caused by what's called childbed fever or puerperal fever. And it's basically an infection of the reproductive tract following birth that leads to sepsis and death. And the physicians had a 14% mortality, but the midwives had a 3%. And they tried changing up where the air was drawn from. They tried to change the smells that were present in the room. They tried to move around, but they always kept running into the same problem. And they basically had to write it off as miasma. Now, just real quick, one problem with the miasma theory is the same problem that we have with the four humors and that theory of disease, in that it lets people off the hook. You can just say, oh, he just had a little more black bile than we expected. That's why the treatment didn't work. Somehow there's bad air coming in from the marsh over here. It's not used so much as a causative explanation as a dodge. And that's one thing you want to watch for, is when explanations for disease are really there just to excuse the fact that it happened rather than explain it mechanistically. Anyhow, this guy, a Hungarian named Ignaz Semmelweis, was working in Vienna at the Krankenhaus, the general hospital, and he realized that the reason there was a problem was that the physicians were bringing disease to their patients. And after you hear this, you're going to be shocked that it was only a 14% mortality. These physicians would attend, they'd come to work, and the first thing they'd do would be go down to the autopsy suite and dissect the bodies of women who had passed away during the night, possibly from childbed fever. So there was a lot of pus, a lot of infectious material. There were no gloves, no latex back then. So this was all done barehanded, without washing their hands. These people would then go upstairs and go bed to bed doing pelvic exams on the pregnant women inside the area. So just the fact that not everyone died just blows my mind. So Semmelweis realized this was likely what the problem was, and he started instituting a hand-washing procedure for people leaving the autopsy suite, where he used basically kind of a bleach solution. It was chloride of lime is what they called it. But basically, he'd have them wash their hands for a certain amount of time, then go up, and lo and behold, the fatality rate dropped to 3%. So, quick poll. Do you think he was rewarded for this or chastised for this? A or B? Yeah, there'd be no story if it was A. Yeah, basically, Vienna was still there was no French Revolution in Austria. Vienna was still very hierarchical. And the fact that he was asking all these upper class, higher status surgeons and OBs to change rankled people. Now, the other thing is, Semmelweis was not a good advocate for his own system. He had a major, genuine insight that could have improved medical practice immediately, but he was a poor communicator. He was abrasive, he didn't do a good job of getting his point across, and when he got pushback from people and told to cut it out, he basically just left, started working at another hospital, instituted his reform there, but it was again dropped because of his abrasive personality. Now, eventually, his colleagues, his younger doctor colleagues, who realized what a big deal this was, instituted hand washing, and it dropped again. But we've got decades of people dying for no good reason because he wasn't able to convince them that this was an actual innovation worth embracing. Now, in no small part, this might be due to the fact that his superiors, who had had patients die of childbed fever their whole careers, might not have wanted to think about the idea that they had been inadvertently killing their own patients this whole time. They had nothing but good intentions, but Semmelweis was kind of, yelling at them like, why do you keep killing your patients? Why are you doing this? Not the way to bring them around to your side, especially if you're the low person on the totem pole in a very hierarchical area. Notes for residency. So Vienna was also a major place of innovation for what we're now going to call histology and cellular pathology, taking that microscope and looking even more deeply at the structures of the body. And we get started with Johannes Peter Müller. So here we got Müller, who basically came up with the idea that tumor cells were not something different. They weren't something new that invaded the body. They were normal cells that went bad. So that tumor cells are cells not of alien origin, but they're modified cells that came from existing tissues. That's a pretty good insight, and he's justly famous for it. But he's more famous for the students that he trained, and you'll notice some of these names sound very familiar. First off, we've got Schwann. Ring a bell? So Schwann basically not only described Schwann cells, the myelinating cells of the peripheral nervous system, he's the person who actually made the observation that all animal tissues are in fact made of individual cells. That was not something people had thought of. Now, he was put onto this by a friend of his named Schleiden, who had made the same observation in plants. And he basically said, hey, you should really think about looking at animal tissues. That might be something that's similar as well. Then we've got Henle. So, loop of Henle. The main thing we care about with him is not his observations and his discoveries, which are good. He's the person who came up with the terminology we use to this day. So next time you're banging your head against stratified, squamous, cuboidal, pseudostratified, you can shake your fist at Jacob Henle. Yeah, some of you are doing it already. Very good. But so it's better than having, if you think about it, it's probably a pretty decent system. It's not the worst. And finally, we have Rudolf Virchow. And I really don't even know where to begin with Virchow. So, have you guys ever had that, you're all pretty smart people. You all got here. Have you ever met that person in your class who's just so unbelievably brilliant that you're happy for them, but you're also a little nauseated at how easy and brilliant they are? Yes? If anybody's telling me you're that person, you better put up or shut up. All right. But even if you think you are that person, prepare to have your ego crushed under the lifetime of Rudolf Virchow. So let's talk Virchow. He became a student of Müller at 22, and then an assistant prosector at the Charité Hospital in Berlin. So early 20s, he's working, doing dissection, doing microscopic investigations. And at 24, he figured out what leukemia was. He figured out that leukemia was linked to an excessive number of white blood cells in the bloodstream of these patients who had a lot of lethargy, weakness, and immune compromise. So that's pretty awesome. He discovered what leukemia is. Then he graduated from medical school. So, clock's ticking, everybody. Clock's ticking. So, he kept on doing investigations with the microscope and gross pathology, and he had to found his own journal just to keep track with the amount of articles he was publishing and putting out there. And that journal still exists to this day, so it's still out there. He wrote a report on the typhus outbreak in a rural area and came to the conclusion that the typhus outbreak wasn't due to miasma or bad water or something we haven't talked about yet, is that poor people are just inherently filthy and that's why they get disease, which was basically what his superiors were expecting him to come back with, was that these, filthy poor people were getting disease because of their lifestyle. He came back and said it has nothing to do with that. It's about the hygiene and the bad water pipes and things in that area. And he then became a major force for political reform and hygienic reform all throughout Germany. And Germany, like Austria, being a very hierarchical society, didn't care for him making waves, and they basically sent him to a smaller hospital. He got a professorship elsewhere. But at 35, the sheer weight of his brilliance and findings and discoveries, one after the other, made him the most famous scientist in Germany, and he was brought back as the director of anatomy and pathology at the Charité Hospital. Now, if that wasn't enough to make you jealous, he's as well known as a physical anthropologist as he was as a histologist and pathologist. He devoted a lot of his time to the study of ancestry, human development, and one great thing about Virchow is he really didn't like the anti-Semitic parties that were active in politics at the time in Germany and Austria. And he did a study on German school children and showed that very few quote-unquote pure German school children had blonde hair and blue eyes. And even more horrifying to the anti-Semitic political parties, 14% of Jewish school children had blonde hair and blue eyes. And when Hitler came to power much later in the 1940s, he constantly railed and just said terrible, terrible things about Rudolf Virchow. So if you've got Hitler angry at you 50 years plus after you die, you've done a good thing. As I mentioned, he was a major proponent of public health. When he got elected to the Reichstag in Berlin, Berlin's sewers are considered to be some of the worst in Europe, and by the time he was done serving, they were some of the best. He really pushed forward public hygiene, clean water, because he realized that's what was a major source of disease. He was a progressive politician. He was actually challenged to a duel by Otto von Bismarck. And there's a couple different versions of the story. One says, yeah, I'll fight the duel with you if we do it with scalpels. And the other version was, okay, I'm going to take typhus cholera, typhus toxin, inject it into one sausage, and then we'll each eat a sausage and see who dies. Bismarck did not take him up on the offer to have the duel go that way. And he was there when Troy was discovered. So, yeah. So if you can lead as interesting and amazing a life as him, I am in awe of you already. So that's the mark. That's the bar, people. Go for that one. Now, next name, we're going back to France now, and you've almost certainly heard of Louis Pasteur. Louis Pasteur was not a physician. He was a chemist and a microscopist, and he used microscopy in chemistry to figure out chirality. So if you remember L and D versions of different molecules, that was some of Pasteur's early work. And he figured out not only that there were those different forms of molecules, but he found out that there were problems with spoiled beer, spoiled wine, and other things like that. And essentially, he was brought in because there was a massive outbreak of bad beer and wine. The fermentation was going poorly, it was getting spoiled, and he looked at the bad and the good under the microscope and saw that there was what we now know as yeast in the good batch, but the bad batch was contaminated with what we now know as bacteria. And he came up with a process. Anybody know the name of the process that is used to gradually heat stuff to kill off the bad germs? Pasteurization. So he did that so that any germs that were in there would be killed, but it wouldn't denature the rest of the food that badly. He also disproved the idea that people had been holding on to for a long time, that spontaneous generation could occur. And if you left rotten meat out, flies would just form from it. He said, no, it's because of these bacteria, these nanomolecules that they were called back then, that are around in the atmosphere. And he would, put a piece of raw meat out, covered versus uncovered, see what happened, then covered in a way that it was open to the air, but with the opening being kind of in a long curly Q, so that dust and other stuff couldn't just settle directly on the meat, and showed that rotting was a result of those bacteria getting access to the food. So he was able to figure out a lot of things that made food safer, beer, wine. He also was able to keep the French silk industry afloat because the silkworms got a disease that turned out to be a bacterial infection, and he was able to help them quarantine the ones that had the disease and keep them away from the healthy ones. And all this because he knew how to use a microscope well. Oh, let me back up. Now, Pasteur is going to come up again, but I just want to also let you know, he, in contrast to Ignaz Semmelweis, was an excellent promoter of his ideas. And we know who Pasteur is, and we know about his innovations because he was an excellent showman. So it may not be the most important thing to be boisterous and always going extroverted with people, but if you really care about something and you have something you need to tell, people don't accept it just because it's a good idea. You have to make them understand how good it is, and that's what Pasteur really excelled at. Now, once it was known that these bacteria could be the cause of different diseases, other diseases started to be characterized as due to certain bacteria. And Robert Koch figured out that anthrax, in this case, jumping ahead, anthrax was caused by a specific bacteria. And he came up with a list of what are called Koch's postulates to show that a specific disease is caused by a specific pathogen and nothing else. And with that work being done, different diseases were suddenly able to be understood to be a result of an infection by one pathogen or another, and shortly thereafter, people were able to come up with inoculations against those once it was known what was actually causing them. Now we're going to jump topics here a little bit because infectious disease is only one aspect of health. Early on, mental disturbances were understood to be due to spiritual judgment or evil spirits infecting the body, which is why we presume trepanation was done, to let the evil spirits out of someone's head. But as people began to understand that the brain was involved, different observations started to occur. And essentially the one that's very famous but completely useless is what was known as phrenology. Now a guy named Franz Joseph Gall thought that, okay, the brain has different mental organs, and that's what he called them, mental organs on the brain. And some people had them developed more than others, and which made them better at some things. And his thought was that those overdeveloped mental organs created bumps on the skull itself that could then be palpated, and you could say, oh, yes, you're very creative because you've got a lump right here, or you've got you're colorblind because there's a depression here, that mental organ is underdeveloped. And so we've got these phrenology kind of busts showing where these different mental organs were located. Now, funny enough, all of his rich, influential clients and all the other phrenologists' rich, influential clients, they seem to only have really well-developed good things going on with their mental organs. And people who were criminals or different, from a different racial background or a different ethnic background were presumed to have deficiencies in these mental organs. And so basically, phrenology was discredited not only because there are no bumps on the skull corresponding to the underlying brain, and your brain doesn't develop into, it doesn't develop hypertrophied areas that impact the skull, but also because it was just simply used as an excuse to justify the status quo. And you can see on the right, the phrenology of people who are considered to be Nordic or Northern European. These were the people everybody thought was so wonderful. They had the best developed phrenology, and as you go more southern in latitude into Africa, it was used as a way to justify treating people of other races as less human or less exceptional, and justify the white supremacy that was being very, very much practiced in colonial era Britain, Germany, France, etc. So that's one of the problems with phrenology. Now, a problem with that is that the brain actually does have localized areas. The brain actually does have regions that are specialized for one thing or another. They don't cause bumps on the skull, but that's true. But because phrenology was so thoroughly disproven, the paradigm shifted to that brain activity is very generalized, and there's no localization of stuff in the brain. And that was the theory for a very long time. But it turns out it's wrong. So phrenology was wrong about most everything, but that localization of brain activity turns out to actually be real. And the first real dramatic incidence of this was known as the Phineas Gage incident. So who's heard of Phineas Gage before? Okay, it's a pretty well-known story, but essentially Phineas Gage was a foreman working on the railroad, and they were doing a lot of blasting of rock to make a track for the rails to be laid down on. And so somebody would move ahead of him, drill the hole, somebody else would put gunpowder in the hole, someone next would put a cotton wad in the hole, and then Phineas Gage, as the foreman, would have an iron tamping rod that he'd pack it down with, and then when it exploded, the direction of the explosion went into the rock rather than up. Well, one day, somebody missed putting cotton in one of the holes, and Phineas Gage's tamping rod hit it, ignited it, and the tamping rod shot through his zygomatic bone and out the top left side of his frontal bone. And this is his actual skull. It's in the Harvard Medical School History Museum. And amazingly, he survived the trauma and didn't have enough of a post-injury infection to kill him. So he survived. But his frontal lobe, especially on the left side, had been really badly damaged, and his personality, his ability to control his impulses had completely changed. Now, the popular telling of this is that he became a wild man who was just rude and used a lot of swearing and couldn't interact publicly with people. It turns out over time his symptoms got better, and he was able to hold down a job as a stagecoach driver in Argentina, of all places. But it did fundamentally change him as a person, and that was the idea that came up to people at that time, is that it didn't cause a generalized loss of everything. It caused very specialized losses. And if you want to think about how miraculous it is that he survived, I spent more time than I care to tell recreating my own Phineas Gage tamping rods. So feel free to check this out after the class. But just imagine this thing going through your skull and landing 30 plus feet away and surviving. So this is available for everyone to play with afterwards. My children are not here tonight to distribute it, so we'll just wait till after. So, localization of brain function continued because of the work of people like Paul Broca and Wernicke. Now, Paul Broca had a couple patients with a similar problem. They'd suffered a stroke, and thereafter they were unable to speak, but they were able to interpret language all right. And upon their death, he was able to do a pathologic examination of their brain and found that the damage was restricted to an area on the left hemisphere, and it's called Broca's area. And when we get to Neuro 1, we'll talk quite a bit more about this, as well as Neuro 2 and 3, that this is the area where language is actually organized and put out there. So people with damage this area, they can conceptually understand it, but the words coming out of their mouth are complete gibberish. Likewise, a colleague of his, Carl Wernicke, had patients that had damage, not there, but a little further up here in the parietal lobe. They had the opposite problem, what's called a receptive aphasia. And in this receptive aphasia, they could not understand the language that others were putting out to them, but they could talk just fine. So they would hear gibberish, but they could enunciate very well. And there are, in fact, people with larger infarcts that would have damage in both Wernicke's and Broca's area, and these poor souls have the inability to understand and generate language. The whole world is just speaking nonsense to them forevermore, and they're unable to communicate that to anyone. So, at this point, what is the brain all about? People knew it was involved in mental activity. It was understood now that there were special areas of the brain that helped us with certain things. But what was the brain actually made of? And two scientists working independently, actually not even collaboratively, they were pretty fierce competitors, were able to share the Nobel Prize because of their findings. Now, an Italian named Camillo Golgi developed a silver stain that he could use to stain the neurons in different sections of brain. And his observation was that the neurons were all interconnected. They were all one big network, continuous with each other. However, a Spanish scientist named Ramon y Cajal did the same technique, but found that he could find different little discrepancies between the individual cells. And he painstakingly drew the different cells that he would find in different areas of the brain. We've actually got what are called Golgi cells present there in the cerebellum. And he said that there were synapses, or there were gaps in between the long stretches, the axons of these cells, and other cells. And the two of them could never quite reconcile it, but their combined observations was enough to let them share the Nobel Prize together. Now, one reason why Ramon y Cajal was not taken that seriously is because Spain was kind of a scientific backwater at the time, so no one was really expecting anything of note to come from there. So he was able to figure out that the cells of the nervous system are separate but communicate by way of that tiny little synaptic cleft. So we've talked a lot about Europe, so it's time to get a little bit of America brought into there. Now, the first real course on what causes disease was taught in Philadelphia by a famous surgeon at the time called Samuel Gross. And there's a very famous picture of him called the Gross Clinic, which you may have seen. And essentially, he was of the opinion that surgery had to be quick because the more the body was open, the more oxygen would cause damage to the tissues, and that's what caused infection. Germ theory was known, and he dismissed it. He said surgery had to be fast, not because there were little bugs that were going to cause disease, but because of the oxygen. But America was doing pretty well in terms of development of medical science, medical training, until the Civil War. And the Civil War was just such a catastrophe in terms of the number of people killed, the damage to the infrastructure, and the just sheer amount of money that had been spent on the exercise, that America really slumped into a several decade depression trying to recover. And this is basically that old-timey medicine that we like to make fun of at that point. Medical schools were largely what are called proprietary. So anyone could claim to be a doctor, put the name on there and let the buyer beware. Hopefully they didn't kill too many of their patients. Medical training was largely run as more of an apprenticeship, where you just hang out with a physician or a surgeon for a while, and after X number of years and you paying your dues, you got to call yourself a physician or a surgeon. The high status physicians who came from wealthy backgrounds would actually go to train in Europe. They'd go to Germany and France and London and go to places where there was a lot more medical innovation happening, and then come back to the United States. The first big jump in training and quality control in the United States happened at Johns Hopkins. This is in Baltimore. And the Johns Hopkins School and hospital were founded by a man who died without children, who was a railroad millionaire, and he basically endowed a great bunch of money for the medical school and the hospital. But by the time he died, the stocks had lost their value, and there wasn't enough money to open the hospital and the medical school. So the wives and daughters of people on the board said that they would go across the country soliciting donations if the board agreed to some outrageous rules, that Johns Hopkins had to be a postgraduate school, meaning you had to have a degree before going there. Sound familiar? Students were expected to read English, French, and German. Okay, you guys got off lucky on that one. It had to be associated with a teaching hospital, or at least a place where there could be clinical observation. And then the medical faculty weren't allowed to practice outside the university system so that they weren't distracted by private clients. They were there to teach the students and work with the patients that were admitted to the university's hospital. And women were to be admitted to the school on the same basis as men. Now, of these 5 items, which one do you think was the sticking point? It was the last one. However, the money was not forthcoming any other way, and so the board agreed to the demands, and they raised the money, and Johns Hopkins opened, admitting both women and men, in 1893. Quick aside, the first osteopathic school opened a year earlier, also admitting women and racial minorities. But you don't ever hear about that, do you? So, yay, osteopathy. So, Johns Hopkins was, however, a major source of medical innovation because of the money and because of the talent and the sheer quality of people that were attracted there as faculty and as students. And so, in terms of pathology, the person who was the chair of pathology was named William Welch, and he was as important as a pathologist as he was as basically a talent scout. he found the other faculty and convinced them to come to Hopkins. And people like William Osler, a very famous person who wrote the first textbook of internal medicine, William Halsted, who we'll have a whole set of slides on when we talk about surgery. And Welch is the one who discovered that gas gangrene is caused by a specific pathogen called Clostridium perfringens. At that time it was named after him, but it's now Clostridium perfringens. And not only were his colleagues top of the top, but many of his students became Nobel Prize winners and went on to do a great many other things. So I think William Welch, excellent pathologist, but there's a certain sort of physician and professor out there who is really good at talent scouting too. So if you happen to be really good at talent scouting, don't dismiss that. Look for where good people would be a good fit and help them find a place where they can really contribute. So, medical training in America being incredibly poorly controlled, incredibly diverse, was becoming a real problem for public health. And in 1910, the Carnegie Foundation commissioned what's known as the Flexner Report. And the Flexner Report basically had a guy named Abraham Flexner go from school to school, seeing what their training was, and making recommendations to the Rockefeller Foundation about how medical training ought to happen. And Flexner basically said that the Hopkins model was the best model and that different schools should adopt that. And the Rockefeller Foundation agreed and put up money for schools to convert to the Hopkins model. And essentially, by the time this was done, you had either adopted the Hopkins model or you'd gone out of business. Now, one quick thing about that, though, Flexner was actually the brother of one of the physicians at Johns Hopkins. So these days, that would have been a horrendous conflict of interest, but back then, no one seemed to care. Now, medical education became more uniform, more quality controlled after the Flexner Report. Laboratory basic science teaching was paired with clinical observation. That became the standard. So these are some good things. You know, the frauds, the charlatans, the snake oil salesmen who were able to just pretend they were doctors and, you know, get away with whatever, weren't really able to do that. There was a lot more regimentation in medical practice. However, the AMA, the American Medical Association, became the dominant voice in medical education and used that to start tamping down on any rivals. And so competitors like osteopaths and midwives were very much shunted to the side. And, you know, midwives are kind of making a comeback now. Osteopathy is still making a comeback. We've kind of come back about 20 years ago is when it really started taking hold. But it was very difficult for anyone who was not working in the AMA MD system at that time to get federal money for any reason. And in fact, MD schools, they decreased a bit from 131 to 66, but DO schools dropped from a small number to an even smaller number. It's unclear whether 6 or 5 made it through at the end. And the funding that was available for research in MD schools was not made available for DO schools until much later in the 1960s. So that's why research at osteopathic schools tends to lag a little bit behind, but we're working on it. Now, last and not least are some things that have happened a lot more recently. So molecular biology and genetics have really recently taken the stage, and we've moved from the humoral theory of disease to an idea that the organs are responsible, to an idea where the tissues are responsible. Then it became the cells that were the seats of disease. And in the 1900s, we finally figured out that there were even smaller things inside the body that could be the locus of a disease process. So in the 1940s, we actually were able to figure out that genes and proteins were connected. Didn't quite know how, but there were proteins that were heritable from parent to offspring. And people figured out that it was this weird cellular junk that was presumed to be just kind of a buffer beforehand called DNA that was actually the place where these changes were kept, but no one really knew how. But basically, linking DNA to genes was done in the 1940s, but the mechanism was unknown until in 1953, Rosalind Franklin did some diffraction studies on DNA and had her research borrowed by James Watson and Francis Crick. Now, the exact version of what that happened is unknown. She passed away from cancer shortly after this happened, so we'll never really know what her side of the story is. But essentially, Watson and Crick used the data she had created to deduce this double spiral nature of DNA and how you have A, T, G, C as the base pairs all along that spiral, and that was the basis of heritability. And in 1989, I actually remember when this happened, the first disease localized to a genetic cause was identified. That's cystic fibrosis. So the first disease known to occur not because of a bad organ, tissue, or cell, but a specific gene occurred. And we've only gotten better and better at localizing different genetic loci of heritable diseases thereafter. So genetics and medicine are still going strong. We've got a lot of different things now. You guys heard of CRISPR? Yeah, the more you learn about CRISPR, the more you get excited. Now, one thing I will tell you in my old age, approaching my 50s, is every time something like this comes along, like the Human Genome Project, other things like that, what you'll hear is, we are on the threshold of discovering the cure for every possible disease and curing everyone. And it never lives up to the hype. However, we always end up further along than we would have before. So even if CRISPR is not the magic bullet we hope it will be for cancer and other diseases, it doesn't hurt to put that out there as a way to get funding so that we do move forward and get progress made. I'm not saying lie. I'm saying when you're putting a grant application together, focus on the positives, focus on the exciting things, be Pasteur rather than Semmelweis so that you can get your good idea out there. And if it doesn't live up to all the hype, I bet it will still wind up doing some good. All right, that's it for tonight, and I'll see you guys on the 22nd for Diagnosis and Treatment. And anybody who wants to play with this in a non-aggressive manner, come on down.